Hey everyone, hope you're all doing well. This is part 3 of chapter 16, and I have been playing with my technology issues, and we'll see how well I do today. Um, Hopefully, getting better and better as the term goes on. Now, this day in particular, because I have to do a lot of drawing. Um, I usually tell students that they should be able to make this drawing in their sleep. Not really going to ask you to do that, but I do recommend that you try drawing it because we're going to end up seeing it a few times for the rest of this unit, Unit 3. And that is cross-sectional anatomy of a spinal cord. So, we talked about I talked about spinal cord levels, and there's, set of nerves going out of each level, set because there's a pair, left and a right. And briefly, I would not mind going back and taking a look at those sections really quick. Okay, these sections. So the spinal cord has all of these little sections here, and a nerve comes out the left and the right. We want to look at one of those cross sections. Actually, we can see it here. This is the slide I was talking about meninges, but you can see a cross section there of the spinal cord. And so get past our lumbar puncture and back to cross sectional. So this cross-sectional anatomy of the spinal cord will also show us how a spinal nerve is formed. So I want to start with just the cross-section, which when I draw it on the board is always asymmetrical, even though it's not in reality. Okay, that looks silly, but that is pretty good. Usually they're not this deep on both sides. The anterior is a little deeper. So this over here, just to orient you a little bit, would be the anterior. This is posterior. And then left and right. So in the center of the spinal cord, it looks kind of like a butterfly. That's what I think. Looks like that on both sides, sort of, pretty close. It's bigger in the anterior than in the posterior. Okay, so that is my spinal cord section. Now I need to label some parts. I don't know if you recall back when I was talking about just nervous system tissue generally, I separated gray and white matter. Oh, that is not good. There we go. Gray and white matter. And gray, hopefully you remember, has cell bodies. So that big part of the neuron, think of it as a processing part, it has the nucleus and a lot of the organelles. And then the white matter was myelinated. I'm going to abbreviate that. Axons. That's why it's white, because the myelination has that, has a lot of phospholipid bilayer. So it's a lot of cell membrane, lots of lipids. It looks white. And in the brain, the gray matter, a lot of it's the cortex, the outer layer. And the inside, the creamy filling is white in the brain. In the spinal cord, it's reversed. In the spinal cord, these areas on the outside are white. These areas on the inside are gray. And in the spinal cord, those gray areas we call horns. I'm going to show you those horns. So in the back here, this little strip here, this is the posterior, I'm going to abbreviate, horn. The anterior horn is this corner here. Anterior, horn. And this side over here is the lateral horn. So you have this extra lateral horn right next to the anterior. And understandably, students are worried about, if I have to label this, where's the anterior, where's the lateral? I mean, there's an imaginary, I guess you can't really see it, a gross level line there. If I asked you to identify either of these horns, I would be clearly in anterior or clearly in lateral. I'm not going to go here close to the border and expect you to definitely tell the difference there. Okay, so I'm going to erase those. Now, I've labeled the, I'm trying to switch colors here. Oh, no, I wanted to be blue, sorry. I've labeled the gray matter on this side, and it would be the same on this side. It's symmetrical, left and right. But I'm not going to label both sides. because that would get just kind of messy. On this side of the cross section, I'm going to label the white matter. So the white matter here, these areas, these are columns. So now your textbook uses the fancy word funiculus. Your lab book primarily uses column. I'm fine with column. It makes sense. So these would be, the axons would look like individual dots in cross section. But if you think going up and down the cord, if we look at an entire cord, so really quick, there's our brain, there's our spinal cord. So think of the columns going just literally up and down. Hopefully you remember what information goes up is sensory, information heading down is motor. And these columns have a combination of sensory and motor. Okay, and I will get to that. What's sensory, what's motor in a little bit. Okay, I'm gonna erase all those things. I've discovered a new way to do this. Oh, and I wanna erase all those little dots. There, okay, so this area here, this white matter, is called the posterior column. Not surprisingly, it's in the posterior. This area up here, sorry, this kind of area right here, not a great arrow, that is the anterior column. And this area, hopefully you're way ahead of me now, is the lateral column. Now, unfortunately, I only have 2 colors in this particular program, but get what you get. Because I would have wanted to draw in a third color. This posterior horn, posterior column to some extent, think sensory. It's not, oops, goodness. It is not. In fact, oops, I have problems. Yeah, it's a mess. There we go. Sorry, my apologies. Okay, in the posterior think sensory, it's not 100% the case, but a lot of sensory information is processed in the posterior horn and traveling up and down in the posterior column. Anterior and actually lateral think motor. So we have our motor and our sensory. Lateral, I'm gonna jump ahead just a teeny bit. Lateral is autonomic, the kind of motor that you don't consciously control. We're going to, just for today, concentrate on the anterior horn and ignore the lateral motor. The anterior motor would be like voluntary, when you consciously move. We also call this somatic motor, which is voluntary. Okay, this is a mess, and I'm going to get rid of it. Yay!